regulation of saliva, particular nervous system like sympathetic, what it does to salivary secretion, parasympathetic, what it does to salivary secretion, and what could have been the prospective questions from that zone. I have discussed all those things. Now one area was remaining, I mentioned last day, that is this area. This is the applied part. So the disease which are connected, there are many other diseases, it is not that only 5 diseases are connected to saliva, but as your 4.2, POI 4.2 says that it is about the secretion and function of the juice. So salivary juice or saliva, these are the particular 5 things. Just 2 lines, 3 lines from individual topic you need to know, and all of this will be very interesting for you basically. So first one is the xerostomia. Do you have any idea what is xerostomia? Now point number 1 is xerostomia. Xero means, xerosis means, anybody having idea? Xerosis? Xerosis means dryness. Dryness. So xerostomia is a condition where there is, stomia means mouth. So xerostomia is the dryness in mouth. It can be due to any condition, but the meaning of the thing and why we are mentioning it over here, it is the decreased salivary secretion. Xerostomia means what? Dryness of mouth, and most likely this is due to the decreased salivary secretion. What could have been the cause? Cause can be plenty. There are many causes which can cause this. It can be maybe salivary glands are destructed due to some other reason. Maybe certain drugs, some drugs that decreases the overall secretion. So salivary secretion is down. That can be one of the condition. There may be some problem with the oral hygiene, poor oral hygiene leading to xerostomia. It can happen. There may be certain metabolic condition, like one very important metabolic condition is diabetes. Even then salivary secretion might be low, or patient may complain of some kind of dryness in mouths. So basic thing is it's a dryness in mouth, it's a reduced salivary secretion, and plenty of condition which can lead to this. Second is cyalorrhea, just the opposite of it. Here what happens, there is more secretion to the saliva. So high salivary secretion. What could have been the cause? Cause can be again plenty. It can be due to certain drugs. It can be due to certain other disease condition. It can be, in old age. It can be for some endocrine reason. It can also be for dental caries. Somebody is having poor oral hygiene. So both can happen if you are having poor oral hygiene. So many causes. What's the meaning? 1 or 2 conditions associated with it sufficient for you at this time. Third one is Sjogren's syndrome. That's a specific, you can see, and there are 2 dots on O. It's not English word. So it is Sjogren's syndrome. This is very interesting. Sjogren's syndrome is an autoimmune disorder. It is an autoimmune disorder. Now these are very early phase of your study. So most likely this may be a new term to you, may not be. Autoimmune means your own immunity, your own body defense system is acting against you. So in Sjogren's syndrome, so there are plenty other autoimmune disorder. It can be various forms of arthritis. অনেকের বাড়িতে দেখতে পারবে, বাড়ির লোকের রয়েছে plenty other forms. Even thyroid disorder, it can be autoimmune disorder. Diabetes, a part of it can be autoimmune disorder. There muscle disorder can be autoimmune disorder. Blood disorder can be autoimmune disorder. So autoimmune is one mechanism by which your own immune system is acting against you. So what happens in Sjogren's syndrome? There are many glands in mostly in our mouth, in our face, there are many glands which secretes various secretion. কি গ্ল্যান্ড আছে এই জোনের মধ্যে? স্যালাইভারি গ্ল্যান্ডস একটা, ল্যাক্রাইমাল গ্ল্যান্ড, টিয়ার যেখান থেকে বের হয়। Sjogren's syndrome যেটা হয়, immune mediated destruction of glands. So this line is important. Here you will be finding your own immunity is acting against you. Immune mediated destruction of glands. And by glands, I do cover a number of glands. It can be salivary glands. These glands might be destroyed. It can be lacrimal gland. So these people with Sjogren's syndrome will be complaining of dryness of mouth, dryness of eyes, maybe dryness of skin. So a multivariate presentation of this disease. You just need to know this is an autoimmune disease which destroys glands, which produces some juice or produces certain important secretion, maybe tear, maybe saliva, and once this is gone or these are gone, you will be facing dryness pertaining to that particular gland. So people might say that I have dry eyes, I have dry, gritty eyes, it's very difficult for me to occasionally see things properly, and occasionally and I need to blink more and more. That will be the complaint of the patient basically. And if you have a dry eye, you will have a feeling that your eye is somehow, it's behaving a different way. The patient will say that. Similarly this, but I find it very uncomfortable when I chew food because the lubricant function is gone. Or suppose he is taking some spicy thing and enough amount of saliva is not coming, again discomfort is more. So he will complain regarding that. So that's the one area. Just how things are happening at this point you need not know. You will come to know in subsequent years, but these things are there. Now these two, one is chorda tympani syndrome, the other is crocodile tears. So crocodile tears most of you have heard of, at least if not in science, at least in literature, you have heard of what crocodile tears have. And we say that crocodile tears means somebody who will get many crocodile tears, right, left, I mean, may not be in this class, but in the society you will get. So basically what is crocodile tears? The tear is coming, but it is not due to grief. What we believe, the tear is just a reflection of grief. You are sad, you are shedding tears. But many people can shed tears at will, doesn't require they might want you to be in grief, that's why they are shedding tears, that can also happen. So all these pseudo tears are kind of crocodile tears. Now how this is at all significant to this scenario? So basically these two areas are problem in nerve regeneration. So these 4 and 5, it is disorder in nerve regeneration. Now let me explain what is that. And also I need to ask you, what is chorda tympani, by the way? Anybody having any idea? Have you heard of facial nerve? What is that? Facial nerve, এটা কি? Facial nerve কি? আমি কোথায় আছি? না আমি ঠিক জায়গায় এসেছি? ফেসিয়াল নার্ভটা কি? এটা জায়গাটার অ্যাড্রেস কি? এই জায়গাটার অ্যাড্রেসটা কি? And I'm not sure whether I am in the right class or not. Facial nerve কি? সেটা উত্তর দিতে হবে তো. What is facial nerve? To these questions who do not know, they are at least smiling. That smile also happens due to facial nerve. এই যে যারা উত্তর না জানলে বোকার মতো হাসে, সেটাও facial nerve থেকে. So what is this? This is the 7th cranial nerve basically. Have you heard of cranial nerve? Have you heard of nerve? Heard of nerve? What is nerve? এরকম করে সবাইকে প্রশ্ন করলে খুব মুশকিল, একজন করে তুলতে হবে মনে হচ্ছে। What is nerve? একজন ভলেন্টারিলি বলো নার্ভটা কি করে? নার্ভ, হোয়াট ইজ নার্ভ? হ্যাঁ, জোরে বলো। আরে তুমি বললে, তুমিই তো বললে। হোয়াট ইজ নার্ভ? Is nerve the same as muscle or something different? আচ্ছা, এই MBBS ক্লাসে যদি নার্ভ জিজ্ঞেস করি আর এরকম উত্তর না থাকে, এটাকে কি আমরা পোস্ট সরস্বতী পূজা সিনড্রোম বলবো? কি বলবো এটাকে? হোয়াট ইজ নার্ভ? যে কেউ একটা বলো নার্ভটা কি? Have you heard of nerve? What is an yes or no? In Bengali, in English, in Hindi, whichever language it suits. Some other language, if you know, nerve কি? Nerve না জানলে তো we can't go on, না? না তুমি বলো নার্ভ কি? তুমি হ্যাঁ, Yellow color, lemon yellow t-shirt, yes. What is nerve? এই যে তুমি এখন হাসছো, এটা ফেশিয়াল নার্ভের ফাংশন, বাট হোয়াট ইজ নার্ভ? Is this a very tough question? What is, I mean, nerve ki? It seems, right? What is face? Now at least you are understanding that this is too easy as a question, kind of that. Anyway, so nerve is something basically, nerve আমাকে বলে দিতে হচ্ছে, this is very difficult here. So nerve basically it's a structure which conducts impulses or signals from a peripheral structure. It can be your face, it can be your muscle. It is conducting the signals back to a central structure. Central structure can be your brain, central structure can be your spinal cord, or it may also be a local central structure. Occasionally these are known as ganglia, and then another similar structure returning from that central structure to the periphery. So that's all about the nerve basically. So facial nerve, remember, it is the nerve of facial expression. So আমরা যে যে এক্সপ্রেশন গুলো করছি, অবাক হয়ে যাচ্ছি বা চোখ বন্ধ করছি, হাসছি, all these are done by facial nerve. Facial nerve supplies all the muscles of facial expression. Apart from that, facial nerve is a mixed nerve. What does mixed nerve means? Mixed nerve means it is a sensory, it has a sensory component. Sensory means বাইরে থেকে একটা impulse নিয়ে সেন্টারের দিকে আসছে. Sensory means from periphery it is carrying some impulse. Suppose somebody is pricking you with a pin. That's a painful stimulus. So the nerve that is taking that painful stimulus and taking it to your brain, this pathway is known as sensory. And you are just detaching, you are just jumping out of pain. So that is the motor response. Jumping out of pain, or just detaching your hand from the pain, this requires some kind of movement. And movement happens due to muscular contraction. And muscles are also supplied by nerve, but this is the motor nerve. Because you see, the sensory supply is inward, from outside to brain or spinal cord. Motor supply is outward, from brain or spinal cord to the muscle. So that's the whole pathway of a nerve. So nerve is basically a neuronal structure. It's very easy. I didn't expect this silence, however. Maybe 10টা বাজলে হয়তো তোমরা আরো জেগে উঠবে, 9টা বেজেছে বলে most likely you don't, জানি না, সার্কাডিয়ান রিদম most likely অনেকে তো জানেই না কোথায় বসে আছে, সেটাও হতে পারে. It might happen, হতেই পারে. This is called orientation, time space person orientation. নাই থাকতে পারে, অসুবিধা. কাউকে না বলতে পারিনি, কাউকে জিজ্ঞেস করলাম কটা বাজে, বলবে এখন রাত. That can also happen, might happen. You are still মানে রাতেই রয়েছো হয়তো, most likely, who knows. Anyway, so that's the whole thing about nerve. So chorda tympani, যেখান থেকে আমাদের এই humiliation started. So basically chorda tympani is a branch of facial nerve. Though by some process, disease process, maybe due to some injury, maybe due to some trauma, surgery, the facial nerve is injured. Or if the chorda tympani nerve is injured, যে কোনো nerve যদি injured হয়, after few months or after a time, maybe after few weeks, it gets regenerated. Nerve যখন regenerate করে, if there is some aberrant regeneration, suppose nerve fibers were supplying to that particular area, during regeneration it is supplying to that particular area, simultaneously some other areas it is also going. That's an aberrant connection it creates during regeneration. Now again we are coming back. What is chorda tympani? Chorda tympani, so far what I have mentioned, it is the branch of facial nerve. Chorda tympani is the sensory part of this facial nerve. Facial nerve not only limits itself to muscles of facial expression, facial nerve is also a sensory nerve. Chorda tympani is the part of the facial nerve which helps us sensing taste. Facial nerve taste sensation ke carry kore, and chorda tympani is the nerve jeta anterior two-third of tongue from their taste sensation ke carry kore. Taste, যে কোনো খাবারের, যে কোনো লিকুইডের we can understand, we can realize the taste via our tongue, যেখানে অনেকগুলো প্যাপিলি রয়েছে, টেস্ট প্যাপিলি রয়েছে, এইগুলো থেকে ফেশিয়াল নার্ভ, the branch of facial nerve name is chorda tympani, it carries out test. এত কথা কেন আসছে এখানে কেন আসছে? বেসিক্যালি what happens, chorda tympani nerve is damaged, after some time it is getting regenerated, there is some aberrant regeneration মানে যেটা হওয়ার নয় সেরকম regeneration হয়েছে. So what will happen, whenever there is some aberrant regeneration, maybe some connection of newly formed nerve is not limiting itself to tongue, it is also connecting maybe some part of the face. দেখা গেল যে ফেসের হয়তো সাবম্যান্ডিবুলার এরিয়াসটাকে সাপ্লাই করছে, or the lower part of the jaw সেটাকে সাপ্লাই করছে. So what happens in such scenario? Again, trauma to facial nerve or part of facial nerve, that is chorda tympani, regenerating in an aberrant fashion. So some fibers are going back to the tongue where it belongs, and some fibers are going elsewhere in the face. What will happen after this? Whenever these patients will eat something, saliva, যখন আমরা খাবো saliva secretion বাড়বে কি বাড়বে না? বাড়বে. Whenever we are eating, salivary secretion is increased. Last day we have discussed that. Whenever we are eating, there will be bolus salivary secretion. You remember? Now in such scenario what will happen, salivary secretion will also increase, and there will be further secretion from other areas of face. এদের ক্ষেত্রে যেটা হয়, whenever they are eating, you can see they are also sweating from other parts of the face. That is known as the chorda tympani syndrome. Why this has happened? Secretomotor aberrant regeneration of secretomotor fibers of facial nerve, which is the chorda tympani nerve, or the branches of the chorda tympani nerve. So what happens whenever they are eating something, saliva is increasing, definitely that should be, but the regenerated and aberrant structures are also increasing the secretion from other places where it has falsely gone. Understood everybody? That is the chorda tympani syndrome. Similarly, crocodile tears is also the similar concept. সেখানে কি হচ্ছে, some part of the nerves which are responsible for salivary secretions are damaged. When the regeneration is happening, aberrant connection now has been developed which are supplying lacrimal glands. So suppose তাদের ক্ষেত্রে কি হচ্ছে, যখনই তারা খাচ্ছে সাথে সাথে কি হচ্ছে, saliva secretion is increasing. Similarly, the aberrant connections, they are stimulating the lacrimal glands, and tear is coming out. খাবার সঙ্গে সঙ্গে দেখা যাচ্ছে তাদের চোখ দিয়ে যেন জল পড়ছে. That tear has nothing to do with our emotion, that is just an aberrant connection. So এই দুটো, if you forget everything, first of all remember that's a problem which have arisen due to aberrant regeneration of nerves 4 and 5. Chorda tympani syndrome, part of facial nerve sends aberrant growth to other parts of the mouth or of the face, whereas in crocodile tears, aberrant growth goes to the lacrimal glands too. So whenever they are eating will lead to increased secretion. Usually it should limit itself to only salivary secretion, but if you have aberrant short circuits other places, you will see that secretion is increased from abnormal places. 4th and 5th are just like that. So here we end this saliva. This is the applied physiology part of saliva is over. Now we will be moving into the next topic, that is the gastric juice. এখন থেকে শুরু করছি. Take your time, in a minute we are starting the gastric juice. এটা হয়ে গেছে তো? I think হয়ে গেছে. তবে নার্ভ কি বলতে হবে, নার্ভ কি বলতে না পারলে তো খুব মুশকিল. অবশ্য বেসিক প্রবলেমটা হচ্ছে নার্ভটা না জানা নয়, বেসিক প্রবলেমটা হচ্ছে কে বলবে, শহীদ কে হবে, এটা একটা প্রথম প্রশ্ন মাথায় ঘোরে. দ্বিতীয় প্রবলেমটা হচ্ছে যে বলা, তৃতীয় প্রবলেম যে বলবো তাহলে নিশ্চয়ই ভুলই বলবো, এইরকম একটা মানে এটাতে কনফিডেন্স আছে যে ভুল বলবো, এটা নিশ্চয়ই বাজে বলবো. কি কি বলবে এখন, বলে কি হবে, এখন তো কিছু বলার নেই, এখন তো আমি বলবো, এত পরে বললে কি করে হবে. দিস ইজ অ্যাবারেন্ট কানেকশন. যখন যেটা হওয়ার তখন হচ্ছে না, অনেক পরে হচ্ছে. দিস ইজ অলসো কল্ড অ্যাবারেন্স. বেসিক্যালি বলবো, তোমাকে সুযোগ দেওয়া হবে, এরপরে বলার জায়গা আসছে. সো সেকেন্ড ইজ দি গ্যাস্ট্রিক জুস. So more or less, this is the outline of stomach. We should not limit to the anatomical discussion to it. Basic anatomy to কিছুটা তোমাদের জানতেই হবে, because even in this class we will be discussing a little bit of anatomy. But hello, yeah. But more about this and the histological structure of stomach, all this you will know during the anatomy class. Anatomy থেকে শুরু হয়েছে, ইনফিরিয়র এক্সট্রিমিটি চলছে, তাই তো? কি চলছে এই মোমেন্টে? হ্যাঁ, লোয়ার লেগ চলছে, তাই তো? লোয়ার লেগ মানে লেগস চলছে, লেগস চলছে তো? এগুলো স্টার্ট হয়নি তো কোনো? অ্যাবডোমেন কিছু স্টার্ট হয়নি? ওকে, গুড। আচ্ছা, ফার্স্ট অফ অল আমি ফার্স্ট ডেতে আই হ্যাভ গিভেন ইউ আ ক্রস সেকশনাল পিকচার, ডে 1, ওইখানটা একবার ব্যাক করো, ক্রস সেকশনাল পিকচার। That is very important. First, day 1, first picture I have given, that was a cross section of the GIT. So stomach, why I am referring you to that? Stomach is a tube. Tube er moto to? It is a hollow tube. Maybe it is a specialized tube. It might be looking like a sac. It is, it is inflated in some part. Top part is narrow, bottom part is narrow, and top part is connected to the esophagus. Bottom part is connected to the small intestine. First part of small intestine, that is duodenum. But still, it's a tube, hollow tube. And any hollow tube in GIT, if you make a cross section, it will look exactly like the same, the picture that I have given. So the picture, if you look at the picture once again, you can see the outer layer is the serosa. Then we are having myoneural layers. So we are having orbic plexus, we are having longitudinal muscle, we are having Meissner plexus, we are having circular muscle, then submucosa we are having, and at the end we are having mucosa. That's the whole picture. And in mucosa there are epithelial cells. So our today's discussion will all the time have this idea, this cross-sectional idea. So what is the cross-section? If I just cut it over here and see under microscope, it will lead to that picture which we have given in the day 1. Remember that picture you can refer to. That's the point number 1. Now coming to the separate parts of the stomach. So if we look at the stomach, there are various parts. We can break it down into 4 major parts, individual parts having individual anatomical and histological connections, histological characteristics, particular group of glands, particular group of cells. We will in this class only enumerate them, and further explanation we will get from the anatomy book. Clear? আমরা এরপরে ফাংশনের দিকে চলে যাব বেসিক্যালি, বাট বিফোর দ্যাট ডেফিনেটলি উই নিড টু ডিসকাস সামথিং। সো দিস পার্ট ইজ নোন অ্যাজ ফান্ডাস। ফান্ডাসটা সবসময়, ফান্ডাসে there will be some gas present. ওই জন্য if we do a straight x-ray abdomen, if you stand in front of the x-ray machine and you just get your abdominal x-ray done or even a chest x-ray done, you will see on the left side, this is my left side, your left side will be on the opposite side, a top area is always black on x-ray. X-ray দেখবে below the ribs on the left side, the top area is always black. That is due to the gas shadow, because you can see that is the topmost part. So gas will always be in the topmost part. If you can see gas on the right side, that is an emergency. Where gas should not be, it is there. That can happen if there is a perforation in the viscera. Otherwise, normally right side, the abdominal x-ray top part is white. Left side on abdominal x-ray top part is black. Black is equivalent to air. Lungs যদি রিপসটা বাদ দেই তাহলে ব্ল্যাক থাকে if you look at the x-ray. Here also the air is there. Where? In the fundus. Now some other parts, this part is known as cardia. This part is known as cardia. This major part is known as body, and this major part is known as antrum. There is one particular structure where things are getting constricted and again dilated. So basically the pyloric area, and this is the duodenum. Various cells you can get in various glands you can get in stomach, various cells you can get in stomach. So we will be enumerating different glands. তাহলে এটা লিখে নাও, glands in stomach. One is oxyntic glands, glands যেগুলো থাকছে, oxyntic glands, cardiac glands, and antral glands. So basically these are oxyntic gastric gland, cardiac gastric gland, and antral gastric gland. So basically three gross variety of glands are present in, you can also say antral or pyloric, okay? You can also say antral or pyloric gastric glands are present in stomach. Three types of glands basically. লিখে নাও। Cells, if we look at cells, usually 6 types of cells but not limited to, আমি লিখে দিচ্ছি তারপরে কমপ্লিট করো, হ্যাঁ লেখা হয়ে গেলে জিজ্ঞেস করো যদি কিছু অসুবিধা হয়। প্যারাইটাল সেল, ECL cell, B cell, G cell. Anything I missed? Mucus cells. Okay. লিখে নাও, কিছু প্রশ্ন থাকলে করো, কিছু লেখা বুঝতে না পারলে সেটা। These are the usual 6 cells আরো আছে, বাট আমরা যেহেতু শুরু করছি we just have to go through like that. 3টা টাইপের gland, 3 types of glands and 6 types of cells. So shortly I will also be drawing the cells on the diagram of stomach so that you can understand where they exactly located. কারো প্রশ্ন ছিল? I think the উত্তর এসে গেছে, হয়তো কিছু বুঝতে পারছিলে না, most likely পেয়ে গেছো anyway. So these are individual cells. ওই পয়েন্টগুলো জাস্ট, এগুলো সেলগুলো, ভেরিয়াস সেল আমি পয়েন্ট আউট করছি, তার আগে জাস্ট এই জায়গাটাকে ক্লিয়ার করো, পয়েন্টগুলো কমপ্লিট করো। আচ্ছা, এনি আইডিয়া অ্যাবাউট এই সেলগুলো কি করে? তোমাদের এখন অব্দি বেসলাইন কোনো আইডিয়া আছে শুরুর আগে? হোয়াট ইজ দ্য ফাংশন অফ প্যারাইটাল সেল? হ্যালো, HCl সিক্রেট করে, এই তো জানো তো। আচ্ছা, চিপ সেল জানো তো? ডি সেল, ডি সেল গ্যাস্ট্রিন। তাহলে জি সেল কি? জি সেল সোমাটোস্ট্যাটিন। ব্রেকফাস্ট করোনি, উল্টে গেছে ওই জন্য। আচ্ছা, আই সেল, আই সেল তো লিখিনি, ছেড়ে দাও। মিউকাস সেল, এটা তো ইজি। There are 2 types of mucus, insoluble, soluble, if you remember. So mucus cell গুলো mucus secrete করছে। What else? ECL cell লিখেছে কি? ECL cell লিখলাম তো বোধহয়। আরো সেল আছে, S cell, K cell, এগুলো আছে, কমপ্লিকেটেড হবে। ECL cell, হিস্টামিন, ECL cell, হিস্টামিন। আচ্ছা, আমরা এবার যাচ্ছি এখানে এই দুটো, these are the parietal cell. This is the mucus cell. Now, what is the function of parietal cell? একটা হচ্ছে secretion of HCl, and the second is secretion of very important intrinsic factor. this intrinsic factor? কি করে? Vitamin B12. Vitamin B12 এ কি হয় এটা intrinsic factor? How it helps in transport and absorption of vitamin B12 till কত দূর অবধি যায়? কোথায় secretion, কোথায় absorption of vitamin B12? What is the location? Ileum. Which part of ileum? Terminal ileum. That means distal. এই তো উত্তর আসছে। তো তোমরা না বলতে পারো নি কেন? তোমরা কি চেঞ্জ হয়ে গেলে এর মাঝখানে? ওকে, সো তাহলে দুটো জিনিস it secretes. One is the axial, the other is the intrinsic factor. এরপরে যেটা আসছে, this is gastrin receptors which are present over there. So when GRP, gastrin releasing peptide, acts on them, it secretes gastrin. Mucous cell secretes mucus. There is a cell which secretes secretin. It's a GI hormone. There is D cell which secrete somatostatin. There is again duodenal G cell which secretes gastrin. And one more cell যেটা এখানে আরো অ্যাড করে দিচ্ছি, there is K cell, and K cell secretes gastric inhibitory peptide. Gastric inhibitory peptide, GIP. Gastric inhibitory peptide. এর অনেকগুলো ফাংশনস আছে। ইন ফিউচার ইন এন্ডোক্রাইন ইউ উইল বি লার্নিং দিস থিংস। So these are basically the whole structure and functional anatomy of stomach. Coming to the nerve supply of stomach, sympathetic supply, parasympathetic supply, and local supply. Sympathetic and parasympathetic is coming from autonomic nervous system. স্টোমাকের সিম্প্যাথেটিক হচ্ছে T4 to T10 সেগমেন্ট। স্টোমাকের সিম্প্যাথেটিক হচ্ছে T4 to T10, আমাকে এগুলো লিখতে হবে না আই গেস, T4 to T10 সেগমেন্ট। প্যারাসিম্প্যাথেটিক হচ্ছে ভেগাস, অ্যান্ড ভেগাসের নিউরোট্রান্সমিটার হচ্ছে ACH। ভেগাসের নিউরোট্রান্সমিটার হচ্ছে ACH। লোকাল সিস্টেম এন্টেরিক নার্ভাস সিস্টেম, আবার সেই পিকচারটায় রেফার টু দ্যাট পিকচার, and there is orbic plexus and there is Meissner plexus, and each of them individually they secrete a number of neurotransmitter. তার মধ্যে you can have VIP, you can have histamine, you can have ACH, you can have substance P, you can have NO. কয়েকটা জাস্ট লিখে দিচ্ছি যেগুলো হচ্ছে লোকাল সিস্টেম, লোকাল নার্ভাস সিস্টেম, তারা সিক্রিট করছে VIP, substance P, histamine, NO, nitric oxide, NO, nitric oxide. দেখো multiple, we can have multiple other neurotransmitter, kind of norepinephrine, it can come out. So multiple neurotransmitters are there which helps in production of these acids. Now coming to the function of the stomach. Next topic হবে function of the stomach. এটা লেখা হয়ে গেছে, আঁকা হয়ে গেছে everybody? কারো হয়নি এরকম? Should I wait for another 1 minute? মুছে দিচ্ছি, হয়ে যাচ্ছি ফাংশন, মুছে দিচ্ছি। This is all about, এখান থেকে যেগুলো আসতে পারে মোস্টলি দি MCQs। এই জায়গা থেকে কোন সেল কোন হরমোনটা সিক্রেট করছে, অর কোন সেল কোন এনজাইম সিক্রেট করছে, দ্যাটস দি MCQ মোস্টলি। ইন This is not very common in university exam, but occasionally we can also get functional anatomy. Describe functional anatomy of stomach. Then this picture, যে ছবিটা আঁকলাম, এটাকে তোমরা বানাতেও পারো. You can, I have just given a very simplified picture. Some functional anatomy, whichever textbook you are reading, you can have further complex thing, you can draw that. এটা আসতে পারে. যদি functional anatomy আসে, description এর question আসে, in that case, a diagram is mandatory. Whether you are making it more complex or less complex doesn't matter that much, but presence of a relevant diagram is mandatory. এই ধরনের কোশ্চেন গুলোতে অনেকে অনেক সময় কোশ্চেন করে যে আমরা তো ছবিটা আঁকলাম, এরপর কি should I write a few lines? Definitely, unless they only say you to draw a diagram and label it. In all other cases, always give a paragraph of writing at least to any diagram in any part or any subject in next few years. এটা খুব ইম্পর্টেন্ট. তাহলে you will be getting close to full marks. যদি শুধু ডায়াগ্রাম দিয়ে বাড়ি চলে আসো, then basically you can hardly get, if the diagram is very vivid and very prominent, very correct, you can go up to 70% max, and a very kind-hearted examiner 80%. But here's a chance of getting up to 100% if you support it with a proper paragraph. Remember this one. Okay. Now we are moving to the function of the stomach. So what are the function of the stomach? আমার থেকে তোমাদের বলা better. What are the functions of stomach? কি কি কাজ করে stomach? Storage. Agree? Storage. Digestion. Agree everybody? Acha. In exam you need to write a few lines. Storage. 2 to 3 liters easily store করতে পারে. Okay? Digestion definitely. কি করে digestion করছে? HCl, pepsin, mainly দুটো. Gastric lipase, 3 নম্বর. So mostly these 3. কিভাবে করছে? HCl is breaking down the peptide bonds, pepsin is acting on it, and further simpler substance of protein is being formed. To some extent, gastric lipase acting on the lipid substances, breaking it down to the simpler substance. Usually triglyceride of the reach করে যায় in case of stomach. Okay? What else? এ দুটো বাদে mechanical mixing of food. Mechanical mixing of food. Why? There will be contraction and relaxation. So whatever food particle is present inside, that is mechanically being mixed thoroughly with the gastric juice, which aids in digestion. What else? Formation of intrinsic factor. Formation of intrinsic factor, definitely, and which helps in B12 absorption. Intrinsic factor, if it is not present, there is a disease, we call it pernicious anemia. Why that pernicious anemia happens? Pernicious anemia is also, again remember, this is an autoimmune disorder. So there are antibody to gastric parietal cell or antibody to intrinsic factor, which leads to destruction of the parietal cell or destruction of the intrinsic factor, leading to deficiency in vitamin B12. যদি vitamin B12 deficiency থাকে, what kind of anemia happens? RBC সাইজ গুলো ছোট হয়ে যায় না বড় হয়ে যায়? ডিফিকাল্ট শুনেছো এটা কখনো? শুনে তুমি তো বলবে বলছিলে, এবার বলো। বড় হয়ে যায়। What does it mean? কি ধরনের অ্যানিমিয়া এটা? ভেরি গুড, ভেরি গুড। মেগালোব্লাস্টিক বলতে পারো, ম্যাক্রোসাইটিকও বলতে পারো, সাইজটা বড় হয়ে যায়। সাইজটা যে বড় হয়ে যায়, এটার একটা ভলিউম, একটা ইন্ডেক্স আছে, সেটা জানো কি? MCV, হার্ড অফ দিস টার্ম? কি সেটা? মিন কর্পাসকুলার ভলিউম। নর্মালি কত অবধি থাকে? ভলিউমটা নর্মালি কত থাকে? We are getting in more and more difficult level. Okay, I'm happy that you could go, get up to there. Okay, acha. So intrinsic factor deficiency, it can happen due to antibody against parietal cell or antibody against intrinsic factor. This is known as pernicious anemia. Sufficient knowledge for your ear. And and who can go further in pernicious anemia will be having high MCV. That's it for you. What else? What other function of stomach? Immunity. Can it help in immunity? Definitely it can help in immunity. দুভাবে করে. The high acid or low pH, it destroys the bacteria. Very low pH is present in stomach, so it destroys the bacteria. What else? It helps in oxidization of iron. So it helps in oxidization of iron from ferrous to ferric formation. Acid যেটা থাকছে, অ্যাসিডটা কি করছে? অক্সিডাইজ করছে আয়রনকে. So ferrous in food is converted into ferric. That also happens in stomach. So more or less, and also acid secretion can activate certain hormone and certain enzymes. So biochemical action is also present. কিছু হরমোন এন্ড এনজাইমকে এটা অ্যাক্টিভেট করে, কিছু হরমোন এন্ড এনজাইমকে it inhibits. So biochemical action is also present. So these are more or less the functions of the stomach. তোমাদের এরপরে যেটা থাকছে যে গ্যাস্ট্রিক জুস, মুছে দিচ্ছি, এই জায়গাটা ইম্পর্টেন্ট, এটাই আমাদের আজকে মোস্ট ইম্পর্টেন্ট জায়গা, যেটা উইল বি গেটিং সাম কোশ্চেন্স, দ্যাট ইজ দ্য ফর্মেশন অফ HCl. Formation of HCl. So this is the oxyntic or parietal cell, suppose. This is the capillary. This is the capillary. And this is the oxyntic cell or parietal cell, and this is the lumen, gastric lumen. So the way we are drawing, if we refer to that, the cells are here. So this is the blood, this is the lumen, this is the cell, the way we are drawing it. If we magnify this, so this becomes the whole structure. কিভাবে হচ্ছে জিনিসটা? Capillary মধ্যে we will be getting two substances. One is water, because in capillary we are having plasma. Plasma is filled up with water. So in capillary we have plenty of water. Another substance that comes from the capillary is chloride ion. So these two substances are here, which will be seeing that these are being used within some time. Okay. So যেটা হয়, HCl secretion এর জন্য we need one molecule of or one atom of H plus ion and one atom of chloride ion. This is required to be present in the lumen. How things are happening? So in any cell, due to metabolism, carbon dioxide is created. And 60% of any cell in our body, 60 to 70% is water. So these two ingredients are plenty in any cell, so as in the oxyntic or parietal cell. Now there is one particular enzyme which whenever presented or whenever is present, this reaction moves into the rightward direction. Enzymeটার নাম কি? Carbonic anhydrase. So this carbonic anhydrase will make H2CO3 carbonic acid, but it is an intermediary which is non-constant. So what happens? It is again broken down into, let us do it here, into H plus and HCO3 minus. So now we have two substances inside the stomach. One is H plus, the other is HCO3 minus. They are in excess. This HCO3 minus goes towards this, whereas this H plus goes towards this. Now try to understand. This area, lumen, is already acidic. Lumen acidic to? Gastric lumen acidic to? pH কত gastric lumen এ? 1.5, 1.2, 0.9, 1.3, whatever. But this is severely acidic. The lumen is severely acidic. Now H plus ion, সেটা তার মানে কি অ্যাসিডিক মানে কি? লুমেনে H plus এর concentration বেশি না কম? বেশি. So what do you feel? Can H plus ion simply go into lumen? No, because lumen has serious concentration of H plus. So we need a transport mechanism where H plus will be kicked out into lumen against its concentration gradient. That's very important. Lumen e jodi ekdom H plus na thakto, H plus will simply go into the lumen. Once again, lumen has very high H plus concentration. Why? Because lumen is acidic environment. Acidic environment means pH is less. pH less means negative logarithm of pH is high. That is H plus ion is high. So very high, million times H plus ion is present already in lumen. The H plus inside the inside the cell can't just simply go into the lumen. So what we need? We need a particular transport, and this transport is an active transport. First of all, try to understand. So there are two types of transport. One is active, the other is passive. So active transport happens with the help of ATP. Active transport happens against concentration gradient. Against concentration gradient. And this is a particular type of active transport. We call it primary active transport. 1 এর উপরে degree sharp মানে this is primary. 2 and degree means secondary. 3 and degree means tertiary. That's the easier way to denote. That's a nomenclature you can remember. That way we do. So primary active transport একটা রয়েছে কোন সাইডে? this side. And this primary active transport তাতে একটা ATPase is working, and this transport is known as H plus K plus ATPase. So that is present on the luminal membrane of parietal cell. This is a primary active transport. Active transports are meant to kick any molecule out of the cell against or inside the cell, whichever direction depends, against concentration gradient. Clear? So this H plus K plus ATPase. So H plus আয়নটা বাইরে যাচ্ছে, K প্লাসটা ভিতরে ঢুকছে, পটাশিয়াম ভিতরে ঢুকছে. Why this is important again? H plus is going outside against concentration gradient. So H plus going outside in a heavy load, it means cell will be losing cations. So to electrically balance it, we need another monovalent cation to enter. So which monovalent cation enters here? The K plus. The whole situation happens with the energy that is coming from the breakdown of the ATP. So we need an ATPase. ATPase is an enzyme which breaks down the phosphate bonds, high energy phosphate bonds in ATP. The energy comes, this energy helps the H plus go out. So what we are getting right now H plus is present inside the lumen. Now one single H plus, if that goes out, simultaneously one bicarbonate comes over here. But again, this is not a very straightforward reaction. What happens? There is something called anionic exchanger. One bicarbonate goes out in exchange of one chloride. So there is an exchanger. This is passive transport. Passive karon ki ekhane kono energy lage na, but it is known as chloride bicarbonate exchanger. Chloride bicarbonate exchanger. This exchanger is a type of anionic exchanger যেখানে একটা anion against আরেকটা anion বেরিয়ে যাচ্ছে. This is anionic exchanger. So overall you can see electrically cell is still neutral. একটা H plus বেরিয়ে গেছিল, তার against একটা potassium চলে এসেছে. So one monovalent cation out, one monovalent cation in. And here one monovalent anion out, one monovalent anion in. So bicarbonate is going out and simultaneously chloride is coming in. This chloride passes through a chloride channel. It doesn't require anybody else. So this chloride is coming out through this chloride channel into the lumen. And what happens to the potassium? This extra potassium again comes out through a potassium channel over here into the lumen, and this again continuously take part in the circular reaction. So the whole thing is like that H plus K plus ATPase is helping H plus to get into the lumen. We are getting one H plus in the lumen. Similarly, bicarbonate is being exchanged with chloride ion which is present in the blood. This chloride is getting inside the parietal cell through the anionic exchanger, Inside the parietal cell, this chloride ion travels to the opposite side of the membrane. There is a chloride channel. Without help from anybody else, this chloride comes into the lumen. Now we have one H plus, net one H plus and net one chloride in the lumen. They just mix up and creates, because this is a physical reaction. These are non-organic basically. So they mix up and creates the HCl. K helps, potassium helps in the circling or continuous circling of the reaction at H plus K plus ATPase. তোমাদের এইখানে এইটুকুই লাগবে, যেটা ইম্পর্টেন্ট এখানে H plus K plus ATPase has a specific name and we call it proton pump. H plus K plus ATPase has a specific name. This is known as a proton pump. Remember, this has come many times in the exam. কিরকম ভাবে এক্সামে আসতে পারে? As a short note, it can be proton pump, it can be H plus K plus ATPase. So the short note if comes, suppose তুমি 5 নম্বরের short note দেখতে পাচ্ছ, you have to give this diagram definitely along with something you need to mention. And it is not over yet. Here some applied physiology part I will be mentioning, that is proton pump inhibitors. এইটা বলে আজকে আমি শেষ করব. What is proton pump inhibitors? That is also another short note. Proton pump inhibitors. So proton pump, if proton pumps are acting more, what does that mean? We will get more acid or less acid? Proton pump বেশি কাজ করলে বেশি অ্যাসিড সিক্রিট হবে না কম অ্যাসিড সিক্রিট হবে? What are proton pumps? H plus K plus ATPase. If they are acting more H plus will be in the lumen or less H plus will be in the lumen? More H plus in the lumen means more acid in the lumen or less acid in the lumen? More acid in the lumen. More acid in the lumen in a dysregulated fashion means more chance of peptic ulcer disease or less chance of peptic ulcer disease? More chance of peptic ulcer disease. So there are certain regulatory mechanism and safety mechanism in place which checks the maximum amount of HCl in the lumen. We will come to that in the next class. But এইটা এখন অবধি তো বোঝা গেছে, যদি HCl বেশি হয় there will be higher chance of peptic ulcer disease. So there are certain molecules, certain medicine which inhibits, which blocks this H plus K plus ATPase. So medicines that blocks, medicines or drugs that inhibit H plus K plus ATPase is known as proton pump inhibitors. They are usually used in gastric ulcer disease. So gastric ulcer disease, or you can say a better term for you will be peptic ulcer disease. Next day we will explain why this is a better term. But these are the medicines that we use in peptic ulcer disease or acid peptic disorders. H plus K plus ATPase. Common drug এর নাম দিয়ে পরীক্ষায় এসছে omeprazole. These are the common drugs. Pantoprazole. Many of you know these names. So these are from this particular drug class. There are lansoprazole, rabiprazole, many such class are there. So I think পরের class time হয়ে গেছে, quickly finish this. Clear? H plus K plus ATPase clear to everybody? This is a short note and explain why. Why lansoprazole or rabiprazole is used in acid peptic disorder. This is a very common explain why question. Formation of HCl 5 marks, short note. একদম মাথায় রাখবে। আর H plus K plus ATPase 5 marks, short note. Mechanism of gastric acid secretion, part of a long question. This can come from anywhere. MCQ তো যে কোনো সময় ফর্ম হতে পারে। I can erase?
Hope the short breaks went well for the pujas. And we are back into anatomy now. Before the upcoming exam for the senior batch, we do not have any off station right now. So till that time we'll be having some anatomy sessions. And we'll be starting this proper anatomy, which you call them as gross anatomy. But before that, we had to finish a couple of things, or a couple of issues related to the topics which is related to genetics. Now we covered the genetics topics like the chromosomal aberrations, chromosomal diseases, and we learned about this chromosomal karyotyping process. procedure as well. So we have a short class to finish this genetics. We are going to talk about the pedigree analysis. So if you have a genetic disease which is running in families, we have to just like understand how this disease is spreading amongst this offsprings or next generation, or how come this disease have coming from the parents or grandparents, etc., etc. We call them as pedigree analysis. So we're going to talk about the pedigree analysis for the day, and we're going to finish off this genetics part with this particular topic. The rest part of the genetics, the topics and the diseases, whatever is there, will be covered in biochem in the second block or second half or third half of the session for you. So we are going to talk about this particular thing. We are familiar with these particular terms. We call them as homozygous, heterozygous. We talk about recessive and dominant. So these are the familiar terms. We are quite familiar with these particular entities. We know about or discuss about this. Then we just like move into the diseases or movement of the diseases in families. We call them as a pedigree analysis. Now, to talk about the homozygous and the heterozygous. So we know that this chromosome consists of the genes, and these genes has a particular spot in the chromosome where they are sitting. The place where this particular gene is located, we call them as alleles. Now these particular alleles which are housing that particular gene are going to express, that means going to just like produce some kind of like a proteins. Now that proteins are coming through the sequence of the events, and these particular proteins, once they are expressed, they are getting some phenotypic characteristics, they are giving some shapes. Like they may be controlling that all parts of the hair color or texture of the hair, maybe skin color and skin texture, etc., etc. So whatever the genes are expressing coming out of here, they have a particular pattern, how they are expressing. Now how these particular genes are located in these particular regions in the chromosome alleles and how they are expressing, based on that we have a couple of terms to understand. So homozygous and heterozygous. So we call this an individual, heterozygous and homozygous, based on identical copies of the gene if it is present or not. So if these particular chromosomes which have the two sets, paternal set and a maternal set, and this particular paternal set and a maternal set in the same allele or same spot of a particular chromosome, say chromosome number 5, in a same spot where you look at this particular space in the chromosome which is known as alleles, in that particular alleles it has the same gene which is expressing, we call them as homozygous. Now if that particular space in this particular allele in a chromosome, they are not of same genes which is present, we call them as heterozygous. So you have this particular chromosomes which has a short arm and a long arm, and one of the set is coming from our father and one of the set is coming from our mother. So for example, we locate this particular positions of this particular gene, we call this particular area as alleles, or kind of like a spot where this particular chromosome is housed. So in this particular area where it is shaded over here, we have a gene which is, for example, A. Now a chromosome which is coming from my father and another one which is coming from my mother, the father chromosome in this allele, say for example, name the allele as X. In the X allele on the father half, we have a gene located in here which is A. Now on the mother half, on the same X spot or same X allele, if we have the same A gene located, we call them as homozygous. Now instead of this A, this is something else. If it is small a, we call them as heterozygous. So this is a kind of like a concept which you have learned in a schools or med schools beforehand. Is coming over here. So these are the space what we have to know about the concept of the homozygous and heterozygous. If the gene in a particular location is the same and identical, we call them as homologous alleles or homologous genes. If they are dissimilar, not with the same thing, they could be like of same size or same expressions, they may be just looking different or maybe just like doing different protein synthesis, we call them as hetero. That means they are not identical. So if it is identical or not, based on that we term this particular thing homozygous and heterozygous in a particular space. call this as this dominant and the recessive genes. So again, coming back to the same thing, we have these two genes which is placed in here, A and A. So again, we are having this particular dominant and the recessive kind of genes. So you have the alleles which could be of a dominant or recessive varieties, which you are going to talk. So each alleles are producing some kind of like enzymes, proteins, etc., which is being expressed as a character. That's why we're talking about. Now in a heterozygous condition, so when we have this particular genes which is of non-homologous, no heterozygous conditions, which is this one, and this is homozygous conditions like this. So if these genes are expressed, one of the gene is expressing, we call this as a dominant gene. And if the other gene is not expressing, we call them as recessive gene. So we have these two particular genes, which is A, small a and big A, like that. So if this particular one chromosome gene which is expressing, say for example, if A is expressing, we call them as the dominant gene. And the other one, say for example, this particular small a, other gene, which is non-homologous chromosome, or maybe homologous, doesn't matter, which is not expressing, we call them as recessive gene. So that means recessive one, that means it is not producing any kind of proteins which is taking part in any kind of phenotypic or any kind of transformations. Whereas the dominant one, which is taking load and expressing and giving some features, either producing some kind of hormones, proteins, or any kind of phenotypic features. So that's how we just like closely associate these two terms as homologous, heterozygous, and the dominant and the recessive, which is based on how they are present and how they are expressing, how they are functioning. So this is the basic idea about this particular pedigree analysis and understanding the diseases, which is genetic basis of the diseases, which we are going to talk hence onwards. So based on that, this is our topic, what we are going to learn as pedigree. Now what is a pedigree? It's a kind of tabular or charted form. Now whenever we are studying an individual disease of an individual of a subject, we are analyzing how this particular diseases is transmitting in the families, from father to grandfather to grandmother to etc., how they are coming and what are the chances of that particular disease spreading to the lower down to this particular family trees. So it's a kind of like a study of an individual documenting the family relationship of that individual. So that's the definition of this particular pedigree. And this particular pedigree analysis we usually do or substantiate in a way so that it becomes a very clear tabular chart form. So that in a quick time we can analyze this particular disease, how they are rolling down in the families. So whenever you are doing the pedigree study, what's the use of this? It provides the family information, how the disease is running in families, and information's mode of inheritance, how this particular diseases is transmitted through these particular families. And then what this particular assistance or treatment can be done to the individual if they are suffering from it. Say for example, a person is suffering from a family disease. Now they are going for a future family pairing, or if you are going for a marriage. So if you have a disease with this particular family inheritance, we advise that particular person to be advised whenever they are going for a mating or whenever they are going for a marriage, they have to make sure the screen through the diseases for the other half or other partner. Now if they are late in doing that, so already they have married and they are planning for children, and then we have to just like screen through how and whatever the chances of that particular diseases from them to be transmitted to their next generation, to their children. And if it is there, then we have to intervene or advise to some kind of devices or to go for some kind of treatments or therapy, or we just like advise or counsel that particular thing. So we call this particular process of halting or altering of this particular family planning as a genetic counseling, which is a separate entity. This genetic counseling is a kind of like advice which you have to go which you have to give to your patients, whoever is coming to your genetic diseases pedigree analysis. So this is a kind of like a thing which is for used for the genetic counseling, and it is easily readable form. Now during this particular pedigree, we usually use the symbolic things. So whenever you are making a family tree or making a family analysis of a disease, we usually use the symbols. Like a normal one, which looks like that, is a male and female have got different looks, a circle and the square or box, and we have this particular carrier of a disease. We just like a point with a dot like that, and we have the diseases part on the other side, and how this particular mating is signified, or how this mating is symbolized, how this particular next generation is made over here. This is like a different charts we use that for analysis of this or understand this pedigree. And then again, you have different symbols or different signs which is been used to just like figure out or create a pedigree chart for the families, starting from the 2 generations up to this particular individual whenever you are making or whenever you are creating one. So this is a pedigree symbols which has been used to create this particular pedigrees, and which is open for genetic counseling, advice, etc., for modification, etc. Now from here we are going to talk about this particular gene inheritance. So if we have a disease, so we have to focus or pinpoint that particular genes which is being transmitted through the autosomes, or it is going through the sex chromosomes. Now on, whatever we are going to talk, we are going to assume or going to discuss the issues of a single gene inheritance. So there may be a disease which may be caused, or genetic disease which may be caused of multiple gene error. So there may be just like one gene involved in chromosome number 5 in a locus X, or maybe just like have a 7th chromosome which has a telomere region, a TX region, which maybe have that same gene which is also have got some kind of alteration. So maybe the person is suffering from 2 chromosomal diseases of that particular genetic inheritance. But here we are just only talking about or going to discuss about the single gene inheritance. We are not going to talk about the multi-gene or several gene inheritance. So we assume the diseases, or we focus on the diseases which is single gene error, and the single gene error inheritance, how they are processing here. So that's the thing we are going to talk. So single gene inheritance diseases, how they are going. And there are 2 forms of this particular inheritance that can be just like transmitted. It may be just like a disease which is because of this problem of these autosomes. That means we have the 22 pairs of the chromosome autosomes. So which may be have one of the particular one gene out of this particular 22 pair genes or 22 pair chromosomes may have a disease. We call them as autosomal diseases. Or maybe they are affecting this sex chromosomes. Maybe X or Y, they may be affected and they are running on it. So either they are X-linked or sex-linked diseases, or they may be autosomal diseases. We have to just like narrow it down. So if the disease is affected on these autosomes, there are 2 varieties of these particular autosomal diseases there. We are going to talk autosomal dominant, if it is genes is expressing, and other one autosomal recessive, which is not presenting in a way, it is a hiding fact. Again, it may be just like an X-linked dominant and X-linked recessive. That could be other ways of the sex-linked disorders. In addition to that, we have a Y-linked disorders. That means that is exclusive for the Y genes only. So that is happening or transmitting from father to boy or boy to his son, like X-linked, Y-linked disorder. There are very few Y-linked disorders are there. So we do not have to just like worry about this particular Y-linked disorders, because we have only XY antigen which is produced by this Y gene, and that particular Y gene XY antigen which is producing a kind of like a defects or genetic like a abnormality, which is like a hypertrichosis, or which is like affecting that like a hearing of the pinna. So that particular disease, apart from that, we do not have any Y-linked disease to just consider out here at this particular level. So either we are having autosomal dominant or recessive disease to think of, or X-linked dominant and recessive genes to think of. So we just go into one at a time to talk about the autosomal dominant and autosomal recessive recessive factors. So take this particular autosomal diseases, which is autosomal dominant. So whenever you are given this particular thing, we have to know about what is autosomal dominant inheritance. So as the name implies, autosomal, that means one gene out of these 22 pairs of these chromosomes, whatever you have, one gene is defective. And that particular defective gene is expressing variety, it's not hiding variety. So it's a dominant variety, that means single gene, if it is error, that is going to create the disease or phenotypic problems, maybe just like any kind of morphology or structural changes, or maybe just like a synthesizing some kind of proteins, which is going to create some problems. So a single gene, if it is present in that particular individual, that is going to express as a disease. Alright, so one gene is good enough to just like express. So we call this particular diseases as autosomal dominant disorder. So single abnormal gene in an allele, whatever it is, out of 22 autosomes, 22 pairs of the autosomes, it is present and it is expressed phenotypically. Now, this can be of like a two varieties. If that particular gene is double dose, that means if that particular dominant gene, if it is present in double dose, that is two genes are there which is expressing, we call this as homozygous state of this particular gene inheritance, and that creates a very severe load of disease. And if it is a single load gene is there, still we have a disease which is a less severe variety of the diseases we are getting. So autosomal diseases, if it is there, if it is coming. Now, what is the inheritance pattern of that particular autosomal disease disorders which is going in? So irrespective of the sex, it is going. So it could be affecting the male individual, it could be affecting this particular female individual, because it is going through this particular autosome. So no link with the sex of this individual which has been transmitted. So if the father have the disease, then the children's, if it is a male or female, just like it doesn't matter, they are going to have these particular diseases. So it is no have kind of sexual orientations on these particular diseases. Now if it is a dominant gene and if it is progressed to this particular next generation, each generation will have a diseased individual. So each individual which has been just like a mating with this particular, having this particular disease, will transmit to this next generation. So if it is a disease is running which is autosomal dominant in a family, the next generation will have a disease. Now depending on if the mother is a carrier or not, the severity of the diseases will be just like at stake. So there will be a milder variety of this particular disease, or maybe severe variety of that particular disease of the autosomal dominant variety. That depends on the mother. If the mother or the fathers or both of them are having this kind of genes and they are making this, transmitting that particular gene which is diseased one to that next generation in a double dose, that means it is in a homozygous state, they have this more varieties of this or severe variety of the diseases are there. Now half of the children will have the diseases and half of them will not be affected, and the children which are not having this particular diseases will not be carried through. So we have this particular pedigree where we have this particular analysis which is expressing out of this. So this is the affected father, whereas capital A and the small a, these are the two genes are there. Mother's is normal, AA. Where this particular large A is there, which is a dominant gene. So this from the fathers, from the autosomal half, two of these are coming, large A and the small a is coming. From the mother, we have getting both of these particular chromosomes which are coming are of healthy varieties. Now as a result of that, if this particular A varieties, which is mixing up with this normal variety of the A, we get this particular affected variety. So next generation, you can see that there is 50% of chances of transmitting to this disease for the next generation. Compared to there, we have this particular other gene which is healthy gene from the father. It is transmitted through this mother, and coming over here, we are getting a normal variety which is unaffected variety. So there is a 50% chance of this particular individual who is diseased in autosomal dominant diseases, 50% chances of this particular individual to be getting a children which is going to be diseased, and a 50% chance which is going to be a normal. Now what is the catch out of this? If you know this particular disease is running in families and it is the autosomal dominant disorders, now we have to advise this particular individual. So whenever you are going to advise whether to have the baby or not to have the baby. So this particular thing, whenever you are going to say that there are 50% chances of getting a normal healthy child with these particular simple things. And there are 50% chance we are going to have a baby which is going to be diseased, just like the father or mother. So that's the advice that you have to just like make to the families. Now, what could the alternative of this particular individual? Say for example, you have located the father is diseased and mother is healthy. So how can you avoid this particular situation with this particular pedigree analysis like this? So this particular thing, a father which is affected with the disease, which is autosomal dominant disease, and a mother which is healthy. Now, if they are going to cut down the entire chances of transmitting the diseases from the father to the son, so what are the best possible way it can be averted, which could be like all the children could be normal? If there are any chances of getting all the children normal here? So we have a 50% chance of getting the disease, right? And 50% is not disease. Now, is there any chance through your counseling method you can advise to the parents or to this particular family there to cut down this incidence rate? Possible or not possible? Not possible. So looking at this, it's not possible if you are going to see. So is there any other alternative ways of replication is there? So here comes with the knowledge which can be done over here is we counsel the patient. There are chances of getting this particular healthy babies, all of them will be there if you just like take this particular A out of this. So we cannot just take the gene out from the father. So rather we can just like replace with a healthy sperm. So that's why we just like recommend this kind of like individual who is suffering with these particular diseases to going for in vitro fertilizations, which is in form of getting the sperms from the donor. So if you can introduce a healthy donor sperm through here, through the in vitro fertilization, we can achieve a pregnancy to the mother which is going to be all normal. So here comes the role of your office or your clinic to advise the patient how can we can achieve or how can it's possible to avoid the disease. So this is what you are learning, this is what you are training for. So you have to just like go for a solution for this, not from this particular individual, because if you are taking this sperm from the father, the same autosomes or same chromosomes will be coming. So the best way or the only way if you are going to just like bypass the diseases transmitted from the father to this particular son or daughter, whatever it is, by not taking the sperm from the father rather than just like going for the donor sperm and going for this in vitro fertilization and going for this particular progeny like this. So understanding the pedigree doesn't stop or shouldn't be there holding this your learning. You have to just come with the diseases, you have to bring a solution or treatment to it. So you have to just like advise what are the possible things that can be done in that particular individuals. So this is what we are learning, the pedigree analysis and the solution to these particular problems, whatever things are coming on here. So this is what we are getting out here as a disease whenever it is mode of inheritance which is coming from the autosomal dominant disorders. So we have a particular disease which is quite deadly in this, we call them as achondroplasia, where we have this particular defects in collagen and that leads to this particular problem of the synthesis of the chromosomes, synthesis of this particular proteins which is been coming out here and giving to this particular formation of the fragile bone, joints are really fragile and this particular limbs or particular individuals are stunted. So we're going to learn a great detail about the achondroplasia in a physiology, in a biochem. So this is an autosomal dominant disorder where it is transmitted through this. So if the individual is suffering from this achondroplasia, we just like a grossly advise to go for in vitro fertilization for donor sperm on these particular next generation reproductions. So this is how it is kind of important for us to understand the name of the disease and understanding the mode of inheritance. So from this autosomal disorders, we are going to the next variety, which is known as autosomal recessive variety. So where this particular recessive variety, we need a double dose expression of the gene to just like express. So here this particular gene which is needing a double dose, that means we need two genes for a particular disease to express. So if you have this particular causative diseases which is AA or AA. So if we have a double dose of the defective gene, then only the disease is expressed. And if you have a single dose which is present as the diseases, say A is disease gene, and if it is single in number, it is not expressed, and this particular individual is known as carrier. So in an autosomal recessive disorder, again the same thing. We have this particular chromosome which is autosome, have the diseases, and only this particular diseases comes into or shows here only from when this particular individuals have the two defective gene which is in a particular allele is expressed. That means both of the parents has to be carrier. So both of the parents should have this particular disease gene in them, and then only the next generation will be getting this particular double dose of the diseases, and then it will be expressed as the diseases. So whenever this particular heterozygous is there, which is kind of like a single load of the genes, there are no diseases. If only have this particular two loads of the diseases, then all genes, then only it's kind of expressed. So we have this particular pedigree analysis which is coming out here. You can see that this is the carrier father and the carrier mother. So both of them are carriers in that particular aspects. So they have this particular gene set, one of the healthy and one of them are not healthy. So this particular not healthy, which is the R gene, and a healthy is the N gene. Now I see that whenever they are having this kind of like a breaking down of this particular chromosomes, only when this RR is matching, they are getting the double dose, which is the affective disease. So out of these particular four, there is a 25% chances of this individual which is going to be transmitting this particular disease to that particular next generation. So there are 25% chances of getting the affected or kind of like the children who are going to be diseased. 75% chances are going to be healthy or carrier. So out of the 75, 25 is normal, absolutely, and 50% are chances to be carrier. So if you are going to analyze this particular facts about autosomal recessive disorder and going to advise the same, the chances of spread of this particular disease from father to mother, if both of them are carrier, there are 25% chances. Like out of 4 pregnancy, 4 of them, there are chances of one of them will be diseased, and 3 of them are going to be carrier or not diseased. So the chances of spread of infection is pretty less in that particular manner of chances of spreading. Now instead of this particular both of the parents, like father and mother, one of them are carrier, other is not, the chances of this particular getting this particular thing also cuts down, also less. So through this particular process, we have to just analyze this particular pedigree, how this particular family is transmitting the diseases from the father to this particular children. So this is a mode of inheritance in case of this particular autosomal recessive disorders. So again, we have this particular comparison of this particular autosomal recessive and autosomal dominant diseases, which we like sometimes we ask about the compare between this autosomal dominant with this autosomal recessive, which is kind of like a dominant pattern, which is single allele gene is present. In case of recessive, double dose as we are mentioning, it is a heterozygous individual which is affected. It is only like homozygous individual in case of the recessive which is going to be affected. There are no carrier state. Here is only where getting the carrier state is. Then chances of infections or chances of this particular disease to be transmitted, which is 50% in case of this autosomal, 25% in case of the recessive. So these are the comparison of the autosomal recessive and autosomal dominant inheritance pattern of this pedigree analysis there. Now, copying of this particular some diseases which is running in the recessive in autosomal recessive inheritance, which is known as cystic fibrosis, which is a kind of diseases which is affecting this particular lungs or secreting capacity, which is affecting this particular lung or pancreas, etc., etc. these particular diseases, which is cystic fibrosis, we have a chances of getting like lung infections or getting chances of this particular pancreatitis or pancreatic infections, which is built there if it is expressed in a double dose or in a recessive manner. Along with that, we have several other diseases like sickle cell diseases, albinisms, and this particular PKU, phenylketonuria. All these are autosomal recessive disorders. So if this particular affecting individuals is having this kind of expression of these particular diseases, we are going to just like warn these particular individuals when they are going for mating or whenever they are going for the child. We should be just like warned off about this particular spread of infections or spread of diseases to this particular disease pattern. Now coming from these autosomes, we are going to this particular X-linked disorders, which is known as X-linked dominant pattern, again X-linked recessive. Now here is a catch about this particular features of X gene. Now in case of the male, we have only one X chromosome, other is Y chromosome, right? So in case of the male, if we have only X, which is diseased, that is going to express the disease. We call this particular X chromosome status in case of the male as hemizygous condition. So in case of the X chromosome diseases, that's what we are talking about. In case of the female subjects and in case of the male subjects, there are two different entities about the sex chromosomes. Now, in case of this particular autosomal dominant and the recessive, one entity which you just like have to understand once you before go to this particular X-linked diseases is there. So we have only one X chromosome in case of the female, in case of the male. In case of the female, we have this particular two X chromosomes. So if in case of the male, if you have this particular X, which is having that particular gene defect, that is going to express. So we do not need a double dose of gene expression of X chromosome expression in case of the males. So in this particular phenomenon, we call them as hemigenetic disorders. So this particular X, if it is a recessive or dominant gene which is expressing, in case of the male, it is going to be a diseased male. In case of the female, if it is if it is recessive disease, we need double dose. But in case of the recessive X-linked disorder, in case of the male, only single gene expression will create a disease. So we call this particular hemizygous state in case of the male. So this is what we are going to elaborate whenever you are going to talk about this particular X-linked inheritance. So X-linked inheritance, just like chromosomal inheritance, we are talking about the autosome and sex chromosome. There are two varieties. One is like X-linked dominant, that means single gene, if it is diseased, that is going to express, it is going to just like get some diseases. So one affected gene can produce the disease, that's the entity. No male preponderance in case of the X, and all daughters of the affected male will be having this particular diseases. So this is what we are getting this particular affected individual. So we have this particular affected father in this particular entity and a normal mother which is XX. So there are no genes whatever is diseased in the mother, rather affected father have got. And you see that this is one X which is diseased, they are going to affect the father as a diseased father. So we call this particular state as hemizygous state. So from the affected father, one X, which is the red color over here, which is diseased X, and it is transmitted to the next generation, splitting into X and Y. And this particular X, whatever it is mating with X on the healthy mother, so that is being transmitted as affected. And from this father, all the daughters, you can see, it is getting this disease. And this particular daughters, all of them will be affected daughter. So because this is a dominant gene, a single gene is going to express in case of the female subjects or male subject, doesn't matter, only one is enough. So this particular affections of father to daughter expression is a kind of like a hallmark for this kind of X-linked dominant inheritance. So compare with this particular father to son inheritance, you see that from the father we are not getting the X, Y is coming and the mother is healthy one. So the X from the mother we are getting is the healthy X, and the Y from the father is coming, which is a healthy Y. Again, so for the male individual, which is going to have the male child, which is going to be normal male child, in case of this particular female child, all of them are going to be diseased. So this is a kind of like a disease of pedigree. We call this particular pedigree in case of this particular X-linked disorders and X-linked dominant disorders. From where the father who is diseased going to transmit all the diseases to the daughters and not giving any disease whatsoever to the sons or to the male childs. So this is a kind of like a X-linked dominance mode of inheritance pedigree chart, which is denoting the affection level over here. Now what I want you to just like consider out here, you see that this particular X, which is that one X, which is the dominant X, in this state we call them as hemizygous state of this X chromosome. In case of the father, whatever, it is coming through this particular individual. So this is a kind of inheritance which is known as this particular X-linked dominant inheritance. There are very few diseases which transmits like this. One of them is known as fragile X. Basically, the fragile X is a polygenetic disease, but it is one of these diseases. So very few, very handful diseases we get is as X-linked dominant inheritance pattern, what is transmitting from father to one diseased and one healthy of X-linked recessive disease. They won't be diseased, not diseased. Rather, they will be called as carriers. So this is what you have to just like understand about this particular X-linked recessive mode of inheritance. So the dose of the diseases, genes which is creating out here. So in this particular inheritance, if you have a disease, both of the mother's X chromosomes should have this gene which is controlling the disease. So double dose expression, also known as this homozygous expression, should be there to make the female diseased. If one of the gene which is healthy and one of the gene is unhealthy, if it is transmitted to the daughters, they are not going to be diseased. And we enter a new terms for this, we call them as carriers. So there are carriers which is the heterozygous state. And in case of this particular disease, if it is coming from the male, compared to here, this one, only one X chromosome which is unhealthy, which is diseased, that is there. In case of the male, which is there, that is expressed. And this kind of expression we already called it as hemizygous expression. And in case of male, only X is creating the disease. So even though they have a healthy Y, we do not have this escape from the disease. So this mode of inheritance, this is a kind of pattern which is important for us to understand out here. So here we have this particular X and Y, affected father, affected mother, you can see. So from this particular affected X, which is father is affected, which is red out here, the mother was or were carriers in this, one of them are healthy and one of them are crossed red. So from the father, this diseased X is coming, and from mother, one diseased and one healthy X is coming. So when this particular individual is going to get down, there are chances of this particular X, which is transmitted through XX, which is the affected female, which is a double dose affected. That means this particular double X, which this particular gene is transmitted, this child, this female child is grossly affected. We call this as a heavy load disease there, if you have a two double dose expression here. Next one is this particular child, as you can see, one is healthy and the other is not healthy. This is not expressing properly. We call them as carrier female. So coming to this male individual, one of the X which is unhealthy, meeting with this particular, one of these, the unhealthy X which is expressing out like this, XX and XY like this, one of these unhealthy X is there, this affected male is affected over here. And Y which is meeting with this particular healthy X, there is a normal child. So if you have a chances of the pedigree of the infection or this particular disease spreading from this particular next generation, the chances of getting this particular infection is in every generation it is there, either in form of disease as the female, they would be either carrier or heavily diseased. So this X-linked disease or disorders, which is like a recessive pattern of inheritance, if it is coming through this particular pedigree, which is kind of like an disease interpretations which will need to be done. So X-linked recessive factors, which is of importance out here, the diseases which commonly we just like a figure over here, which is known as hemophilia. So hemophilia is a kind of X-linked recessive disorder which transmits from the families, from this particular father to daughter or daughter to just children. And this particular inheritance pattern, which is you have to just like express out here. So the homozygous female, which is grossly affected in this particular case. So this particular heterozygous females, we call them as carriers, and this particular hemizygous, which is a 1X chromosome in case of the male, they are also affected in this particular individuals. So this particular entity of the spread of infections or spread of diseases from one generation to another generation through this particular X-linked disorders, which is like either dominant variety or this recessive variety, which matters to us on this particular individual cases. And again, if we have this particular diseases which is coming over here, the genetic counseling or the advice that we have to make out here. In case of this particular genetic X-linked disorders or recessive disorders, the best thing to just like counsel the individual, if they are going for any kind of children's in future, just like the best thing is that going for artificial process of replications, which is known as in vitro fertilizations, and which is going to be just like either picking up with the donor sperm or either going for the donor ovum, which is the solution for this to eliminate the disease, so that this infections or these particular diseases is not transmitted for one generation to the another generations. So that's the genetic counseling part that we have to make. So this is all about this particular pedigree, the male and female individual X-linked, autosomal recessive and autosomal dominant varieties of this particular pedigrees, which is single gene or single modal inheritance which is going through, and this counseling, which is known as the genetic counseling. If you have to make any, we have to just like counsel this particular individuals, either go for this like in vitro fertilizations or artificial reproductive techniques, we call them as ART, and the ART composed of two varieties. That means this particular individual ART so we call them as artificial reproductive techniques. So this particular artificial reproductive techniques is kind of like could be in vitro, that means it's going to be inside this particular lab. So the best solution is if the mother is having some kind of diseases, we call them as ovum donor, and if the diseases is transmitted through from the fathers, we just like go for the sperm donors. Now coming to this particular carriers, if the family have got carriers, either mother or this particular father, so any of this will be solving the purpose. If both of them are carriers, then we have to go for this particular ovum donor and the sperm donor from the two separate entities. So if we can eliminate one factor at least, so we can just like have a ovum donor and the sperm donor, and we're going for this particular artificial reproduction under microscope, and after that we are planting this particular ovum or zygote into this particular utero, then we're going to have this particular offspring which is disease free, kind of like we can eliminate that particular genetic diseases from these particular generations. So this is what the best we can offer to particular individuals who are the families who are suffering from this kind of like a genetic disease which has this particular pedigree pattern. So once we are going through this thing, whenever these couples are coming for the counseling, we have to just like see for whether it is autosomal diseases or the sex-linked diseases. If it is autosomal or sex-linked, if it is figured out how extensive this particular disease is running in the families, that's what you have to go for the pedigree charts. Now once you have this particular pedigree chart created, you have eliminated the chances of spread of infection or spread of this particular diseases, which is 50%, 20% or carrier rate, etc., depending on the mode of inheritance, how they are coming. So once we have figured out, we have to give a treatment or solution this individual. So the solution based on this, which is going for the ART. Now the ART can be just like based on this. If this particular male is individual which is having this particular disease which is carrying through, then we have go for this. We have to eliminate the sperm from the male. We have to just like take for the donor sperm. If it is coming from the mother end, we have to go for the ovums or picking up the ovums. So this particular entity, based on that, we just like do this fertilization in the lab through this artificial reproductive techniques. Then we are producing the zygote, and then we are planting the zygote inside this utero after creating proper environment, and then we are just like growing the zygote, which is going to be this like a next generation, which is going to be disease free rather. So our part of this particular class is not to just like go to understand this pedigree along with this just like understanding and stopping it where how this disease is coming, rather to bring into the medical part or the treatment part of this ART, which is producing a solution to it. Alright, so this is all about this particular pedigree analysis and the genetic counseling or related part. The genetic counseling in detail and ART will be covered in the biochem sessions. In that aspects, we'll be learning that part as well.